Good day, everyone. My name is Eryn Bugbee, and I'm a third-year undergraduate student in life sciences conducting summer research in the McGuigan Lab. Today, I will be discussing my work on a literature review, Bridging the Divide, Engineering the Tumor Microenvironment. The leading cause of death in Canada, cancer has proven itself to be an elusive and cunning opponent. To better support the staggering number of those affected, many therapeutic methods have been created to manage and treat cancer. In the recent years, the options for cancer therapy have grown. Immunotherapy is the employment or targeting of the immune system to treat neoplastic conditions. While immunological approaches have revolutionized the treatment of some cancers, such as Hodgkin's lymphoma, immunotherapy is no stranger to failure. Unfortunately, immunotherapies targeting aggressive cancers, such as pancreatic ductal adenocarcinoma, have had little success. A significant factor in the failure of immunotherapy in these unreceptive forms of cancer is the tumor microenvironment. The hostile tumor microenvironment, TME for short, is complex and composed of a variety of players, including circulatory networks, immune cells, stromal cells, and the extracellular matrix. The coordination between them often leads to the resistance to therapies through mechanisms such as the promotion of immune cells that suppress inflammation. The current field of immunotherapy relies on models to test their success prior to any sort of practical use. A pillar of immunological research is the in vivo murine model. Mouse models are able to mimic our own bodies much better than traditional cell lines. However, mice are a flawed model and lack key features of human systems. This leads to the demand for models that accurately shows human complexities. In order to create effective immunotherapies, the in vitro model should well reproduce the tumor's complex landscape. Creating a good in vitro model requires a strong understanding of immunological features along with the facets of engineered models. The purpose of this review is to present a comprehensive background on the major facets of the tumor microenvironment and provide 3D in vitro models to probe the effects of these features. Creating a literary review follows a straightforward workflow. First and foremost, the creation of a review question to guide the subsequent research. Following this, multiple search strings were created and used to explore a variety of databases such as PubMed and Scopus. Literature of interest was screened for relevance with a two-step process of first viewing the abstract and then the rest of the paper. Lastly, the relevant information was extracted and compiled into a functional review. Through this process, the selected immunological components were placed into the following categories: immunogenicity, metabolism, tumor architecture, and hypoxia. Immunogenicity describes the way your own immune system interacts in the TME. Depending on the type of TME, certain immunotherapies may be more effective or completely ineffective at treating cancers. The TME can be broken down into two immunological phenotypes: hot tumors, which are T-cell inflamed, and non-T cell inflamed cold tumors. T-cell inflammation is the infiltration of T-cells into the TME, along with the increase of cytokines that boost the immune response. Hot tumors have a higher number of mutations in their DNA, allowing them to be recognized and targeted by the immune system. In contrast, however, cold tumors have a deficiency in surface proteins that aid in immune recognition. Melanoma, lung, and liver cancers all typically have hot tumors, whereas cancers such as pancreatic, glioblastomas, and prostate are often immunologically cold. A promising in vitro model to explore tumor immunogenicity is organoids. Organoids are three-dimensional cultures of a heterogeneous mixture of cells, similar to the variability found in our own tumors. These cell lines can be cultured from patient tumor samples to better represent the host microenvironment. A recent study by Dijkstra and colleagues developed a co-culture model with tumor organoids and immune cells. This model serves as a systematic platform to probe the recognition of tumor cells by the host's immune system. Moving on to the next feature, under cancerous conditions, the TME undergoes a dramatic metamorphosis in metabolism. The three primary displays of metabolic reprogramming are glycolytic shift, lipidomic remodeling, and altered amino acid consumption. The Warburg effect is a hallmark of cancer metabolism that occurs when cancer cells shift to anaerobic glycolysis, resulting in a significant buildup of lactate and subsequent acidosis. Lipidomic remodeling includes the de novo lipogenesis of higher quantities of saturated fatty acids, which allows the tumor to be more resistant to chemotherapies. Lastly, tumor cells exhibit a higher consumption of the amino acid glutamine, which has been shown to favor immunosuppression. Metabolism is difficult to analyze in vitro as it requires rapid collection for metabolite analysis to be conducted. Usually in the past, the standard for in vitro metabolomic analysis is 2D cell cultures. The McGuigan lab at the University of Toronto recently developed the first three-dimensional platform for analyzing metabolic gradients. Tissue roll for analysis of cellular environment and response, TRACER for short, is a paper scaffold embedded with tumor cells rolled around an oxygen-impermeable core. This generates oxygen and metabolite gradients that successfully mimic those found in vivo. After a set duration of time, TRACER can subsequently be unrolled in under a second and rapid snapshot analysis can be conducted. On to the next microenvironmental feature. The intratumoral landscape is quite intricate and diverse, with its components acting in concert to maintain the cancerous environment. Each TME is comprised of a mutual set of components including stromal cells, the ECM, along with vascular and lymphatic structures. Each component plays a role in recruiting and inducing immunosuppressive cells. There's also crosstalk between these components. For example, stromal cells often will induce the ECM to promote tumorigenesis. A set of useful tools for replicating tumor architecture in vitro are hydrogels. A network of polymers saturated with water, they can simulate structural components of the in vivo ECM. There are both natural and synthetic types of hydrogel that can be made into a variety of configurations. Another in vitro model developed by the McGuigan lab uses a hydrogel matrix in a culture platform. Gels for live analysis of compartmentalized environments, or GLANCE, explores the interactions of cancer-associated fibroblasts in the tumor stromal interface. What is unique to GLANCE is that it allows you to view cell invasion under a microscope in real time. The use of hydrogel in this model offers a sophisticated mimicry of the tumor microenvironment. On to the final feature of the TME. A key regulator in the tumorigenic process, hypoxia has created a name for itself in the cancer immunology world. Due to the location of blood vessels in the tumor, tumors will often experience oxygen gradients, with the innermost tumor cells being the most hypoxic. The HIF family of transcription factors exerts an assortment of effects on its host cells. They are responsible for the induction of several key pathways, such as VEGF, which promotes the formation of new blood vessels, or angiogenesis. Each section of the TME is heavily influenced by hypoxia, making it a crucial piece of the in vitro puzzle. To study hypoxia, the use of microfluidic devices is highly advantageous, as there is the ability for both spatial and temporal regulation of oxygen. Microfluidic devices can induce oxygen gradients with oxygen scavenging chemicals that deplete oxygen, gas supply channels that control oxygen levels, and the use of oxygen impermeable materials that can be designed to regulate oxygen flow within the device. A microfluidic device was developed by Funimoto and colleagues to control the oxygen levels of a 3D cell culture using polycarbonate, a film with a small oxygen diffusion coefficient. This device showed the unique directional migration of cells under the hypoxic conditions in the third dimension. Here I have provided a summary of the selected microenvironmental factors along with the models discussed in this presentation that reproduce them in vitro. It's important to note here that different models have different advantages and disadvantages for modeling certain factors of the TME, as well as some models can be applied to multiple factors. This table also highlights the unique approach to metabolic analysis that TRACER provides over other models previously discussed. In conclusion, the tumor microenvironment provides a variety of barriers to immunotherapeutic success. It is important to have a mutual understanding between the TME and current and future in vitro models in order for successful models to be created. This information is useful in the future to guide the work being conducted in the McGuigan lab to probe immunological features in TRACER. In conclusion, fostering communication between immunological and engineering knowledge is important to develop successful therapeutic treatment and to better serve the future of medicine. Thanks for listening. And if anyone has any questions, here are my references, and I will open up the floor.
Thank you, Eryn, for your presentation. Now I would like to address judges and ask if you have any questions. Yeah, so you can unmute yourselves.
Okay. All right. Hi, Eryn. Thank you very much for that presentation. I found it's such a challenge to bridge the engineering and the life sciences world. So what would you say has been your interaction with engineers, and what have you learned from your experience with them?
Totally. I think that's a really amazing question just because I have been able to experience it hands-on throughout the summer. I'd say the one thing that I didn't realize coming into the engineering world is just, I guess, how many similarities there are, but also just the different ways that we look at problems. Because normally, someone with a biology background will look at something and be like, okay, I'm just going to go out and find this. It's already here. And I'm going to use this model or this system to explore an interaction that I'm looking for. Whereas engineers will look at a problem and be like, I would like to make that myself. And they'll take past research or other people's models and refine it to where maybe they're more looking towards taking it in a certain direction, which I think is really interesting because you're not always going to find a model or a device that's perfectly suited to exactly how you want to research.
Mm-hmm. Mm-hmm. Very good. Thank you.
No worries.
Hello, Eryn. Thank you for that presentation. This is Sharon. I wanted to go back to the slide where the summary slide where you had the 4 models and
Yes.
the applications. So hypoxia ticked all the boxes, and so that immunogenicity. Where do you think, which one of these do you think is furthest advanced, is the furthest in advancement, and which one you think has the best applicability in terms of the near-term solving of the problems that we have?
That's a great question. Thank you for asking. In terms of immunogenicity, just for the fact that it is usually so heavily used by immunologists, the most likely model that they are going to use is an organoid co-culture, as I discussed in my presentation. Papers, like all the other models, do show it, but they just aren't as popular, mostly just based on accessibility to immunologists, which is one of the reasons why I wanted to create a presentation like this, because a lot of immunologists won't look at a microfluidic device and say, okay, I can use that to probe immunogenicity, whereas in reality, it actually is a perfectly fine mechanism and just needs a little bit more work on it. And in terms of hypoxia, personally, I've found that the work done with paper-supported models is just a little bit further, and that's another thing that Tracer is quite good at, is they can model oxygen gradients and hypoxia. And there are many other paper-supported models that came after the original one published in the early 2000s. But I decided to discuss microfluidic devices because I wanted to give a little bit more of a well-rounded view into some in vitro models. And in terms of what I would say is the least good of the 4 in vitro models, the organoid one does possess hypoxic gradients, but there isn't a lot of ability to regulate it unless you put it in a hypoxic chamber where you're not actually seeing a gradient, you're just seeing like the whole environment is hypoxic, if that answers your question.
It does. Thank you so much.